structure of viruses. And it's a very fascinating topic, so I'm excited to be here today. I'm not a teacher, so I might be sloppy, and I need you all's help with the class, so interrupt me, ask questions, and there are some difficult concepts that I want to explain, so let's see how that goes. So let's start by taking a look at the viral infection. So what's going on here, we have here a virion here in pink that has found a cell that can infect. So how does it know that it can infect that cell? Well, the surface of the virus, the capsid, is specific to the surface of the cell. So it's recognizing the cells through this capsid, and it's making the genetic material to go through the membrane inside the cell. So now this genetic material can get picked up by the ribosomes. So viruses can use different types of nucleic acid to encode their information. So what would you think that nucleic acid is? In the back?
RNA, obviously.
Yeah.
Translated directly.
Yeah, so viruses that have positive RNA, they have signals in the RNA that make them recognizable by the ribosome. So this genome is directly recognized by the ribosome, and now the ribosome is producing the viral proteins. So we have here this protein in orange, that is the polymerase, that is going to start making copies of the genome. And then as the viral cycle progresses, we see an accumulation of all these pink blobs that are the capsid protein. So at some point, all these blobs are going to interact with each other with different chemical interactions, and they're going to recognize the RNA and they're going to encapsulate it. So this is a process we call assembly, and so you can tell here the RNA is kind of twisted, so it's a single-stranded molecule, but it has complementation between different pieces of the sequence, so it has a lot of secondary structure. So we depict the genome as a line, but it's actually more like a ball of yarn. So now we have all these new particles that we call virions that are going to go outside the cell, and they're going to go infect a new cell. So now the genetic material is protected by this protein, by this shell or coat or capsid. So there is something that's common to all viruses, and that is that the capsid is going to be made by different subunits. And this has a very simple reason. It's impossible to make a protein that is larger than the gene that is going to encode for it. So we have 3 nucleotides that are going to make a small amino acid. So the larger the gene, the bigger the difference is going to be between the protein and the side of the DNA, or the RNA in this case. So in this lecture today, we're going to see the methods that we use to study virus morphology, the types of structures that we can find in the viral world. We're going to see what we understand by icosahedral symmetry, and this is where things are going to get more complicated. We're going to see what quasi-equivalence means, the triangulation that's going to give the different T numbers. This is a way to describe how these different capsid units are going to interact with each other to form this big geometric structure. We're going to see some examples. There are also viruses that look like filaments, so that those have helical symmetry and not icosahedral symmetry. Hopefully we'll have time to see how viruses can assemble and disassemble, and some viruses also have an envelope that it's basically a membrane that they catch from the host. So viruses come in all sizes and shapes, so we find icosahedral or close to spheric viruses. We find some filaments like tobacco mosaic virus. We also find bullets. And then we also have giant viruses. So this one is in color because we can see with a light microscope, but the rest of the images comes from an electron microscope. So the electron microscope is a good tool for studying virus structure. So the main difference between an electron microscope and a light microscope is that we use electrons instead of photons, just because we want more resolution. So this is a FEI technique that we have in SRTC, Science Building 1 in the basement, and we use this machine frequently to look at our samples. So we put the specimen here, that is our sample, in a tiny copper grid, and then there is an electron gun on the top that's going to shoot a beam of electrons, and different magnets are going to act as lenses, and then everything is going to get projected down here, and we can get the image from there. So how do we stain our samples? We can use negative stain that is pretty simple. This is the only one I'm familiar with. So why it's called negative stain is because we don't actually see the sample, we see the shadow of the sample. So we use a solution that is electron dense, like uranyl acetate, for example, and that's going to accumulate in the little holes in the structure, and also between the surface of the grid and the particle, and that's how we get these images. We have the electrons here; they are not going to pass through these electron dense parts, and then we get that image. We can also have an infected cell, for example, infected tissue, and we can make very thin slices with an ultramicrotome, and then we can stain that and visualize, and eventually we can see different particles there. Then there's another technique of electron microscopy that's called the cryo-EM, and this is basically because we're using frozen samples. These allow us to get more resolution, and this is a machine that is down at the waterfront. Steve Reichel uses that machine. So how can we get a model of the structure of the virus just by getting these bidimensional images? Well, we can do single particle reconstruction. So if we have a sample and we have a big number of these bidimensional images, we can pile them on top of each other, and then we can use computational tools to build the three-dimensional structure of the virus. Another way that we can study viruses, viral structure, is X-ray crystallography. In X-ray crystallography, what we need to do is we need to get our sample crystallized, and that's basically having all the particles arranged in a regular way in a small crystal, and then we have that crystal here. We shoot a beam of X-rays through it, and then all these particles are going to cause a diffraction in the beam that we can detect. And that is going to look something like this, which doesn't tell you much unless you have, again, the computational tools to make it make sense. So from these, you can have an electron density map. And if you know the sequence of what you're looking for, you can try to fit that into the electron density map, and then you get a protein model. And then if it makes you happy, you can actually 3D print that model and have it in your hand, like we have all these viruses. So we can see in very general terms different types of virions. We can see spherical virions like this one, and these ones have icosahedral symmetry. We can find filaments, and these have helical symmetry. And then there are also complex viruses like FHD4, and this combination of these two plus variations plus different elements to form this virion, you need more than 20 different proteins. So kind of challenging the idea of just using one subunit to make this big thing. Then we can find variants of these different ones. Like, for example, if we look at the head of this phage, it's not a sphere, it's not a icosahedron, it's slightly elongated, and that's because it has like an additional feature that is going to give it that shape. I have here a really cool example as well. This is called a Gemini virus, and this is basically two spherical capsids put together. And then we also find viruses that are enveloped. They have a membrane around them. So we call naked when they don't have this envelope. So let's see what principles are common in virus structure. Well, we can apply the His principle, keep it simple. Viruses are really good at minimizing and being very simple in the way they operate. So they always try to get very small, and they use different strategies for that. Genetic economy is what I was telling you about before. The nucleic acid is going to be too big for having a single protein to protect it, so they need to make different subunits that are going to assemble together. And in some cases, the capsid gene can make up to half of the genome. And because of the difference between the size of the protein and the size of the nucleic acid is larger as the nucleic acid is bigger, we're going to be needing more and more subunits as the genome gets larger. Then we find symmetry in most of the cases. It can be icosahedral or helical. And then we have a paradox as well. We want the virus to be very stable and resistant, so when it's outside the cell, it doesn't fall apart. But it also needs to get the nucleic acid out once it's inside the cell. So on one side it's very resistant, on the other hand, is very easy to open, and that's what we call metastability. It's a virion is a metastable structure. So let's talk a little bit about icosahedral symmetry. So here is an icosahedron, and we find different axes of symmetry in this structure. We find, let's start by the fivefold axis. That would be the vertices of the icosahedron. So that means that if you rotate this, 72 degrees, that is basically 360 divided by 5, you find the same image. Then there is threefold axis of symmetry. That would be the side of the icosahedron. So if we rotate 60 degrees, 120, we find same image. And then we have the twofold axis of symmetry, that is going to be the edge here. And then we need to rotate 180 degrees to find the same image. So viruses find this symmetry when they assemble their capsids. And here we have an example of, I would say it's a fictitious, it's not a real virus.
Question?
Yeah, sorry.
I was just wondering, do the capsids retain their shape after the genetic material is released out of them?
So it depends on the virus. Some viruses, they just fall apart, so the different subunits just fall apart because the environment in the cell makes all the interactions to stop, and they fall apart. But others, they just release their genetic material, and honestly, I'm not sure what happens with that empty capsid. So in this particle, we can see the different axes of rotation or axes of symmetry. We have here the fivefold symmetry axis, and we can see that in each of the sides of this pentagon, we have a different subunit. Then we have here would be this is the side of the icosahedron, and here we would have the three axes of symmetry, and here it would be the two axes of symmetry. What we can see here is that each of these subunits is interacting with the rest of them in the same manner. But the interactions are going to be different in each of those axes of symmetry. So if this is like a hand, we can see that the wrist is interacting with other wrists in the fivefold symmetry axis, but then the fingers are going to interact with each other like this in this axis. The same with the twofold axis. So at the end, what we have here is a particle that has 60 different subunits. And why 60? Well, we have 5 in each fivefold axis of symmetry, and then because this is the vertex of the icosahedron, there are 12 of them in an icosahedron, and then the total would be 60. Is this clear? Do we have any question? Sometimes you can get confused because what you're seeing here is a pentamer, and you can be like, well, what I see there is not a icosahedron, it's a dodecahedron. So what I want to point out here is that they both have the same symmetry. So dodecahedron is basically truncated icosahedron. So same with here. What do you see here? Do you see an old lady? Who see an old lady there? Okay, you can see an old lady. This is the nose and this is the mouth, but this can be also a young lady looking back. The same with these structures. So yeah, you can see a dodecahedron, but let's stick to the idea that this is an icosahedron because of what I'm going to explain next. But I guess what I want to point out here is that when we talk about icosahedral symmetry, we are not talking about viruses being an icosahedron. They just have icosahedral symmetry. It's not the same. So we have seen how 60 subunits can form this icosahedral capsid, not icosahedral capsid with icosahedral symmetry. But when we look at the viral world, we find a big diversity in virion structures. And here we have different capsids separated by color based on how many subunits are going to make each of those capsids. So we look here at the number of capsomeres, that's how we can call these different subunits. We see that the ones that have 60 are just the red ones. So what happened to the rest? How can we make the rest of all these capsids and have this difference in sizes? Well, you can also observe something here in the numbers, is that they are all multiple of 60. So how is this possible? What's the geometry that is behind the assembly of the rest of all these capsids? Well, there were two scientists that were trying to answer this question in the 50s and 60s, and those were Caspar and Kluge. They were buddies with Francis Crick and his buddy that I'm not going to name, and Rosalind Franklin. I've heard that Caspar had an affair with Rosalind Franklin, and they were trying to figure out what's going on with these capsids. And they came across the work of this designer and architect. It's called Buckminster Fuller. I don't know if he's still alive. I don't think so. Well, he was the inventor of these geodesic domes, like the one in the expo in Montreal. What he was doing is these big structures that is basically putting triangles together in an hexagonal fashion and making them into a big sphere. So how could he do this just with hexagons? Because you put hexagons together and what you get is basically a flat surface, like we can find different ones in nature. Well, he found that you can put curvature into these patterns by introducing pentagons. So here, for example, I could find a pentagon there. I couldn't find any other one. So you can put pentagons in this hexagonal grid and then make a sphere. This soccer ball was also inspired by Fuller. And here what you have is all these hexagons, and then you put pentagons in between and you get a ball. So it was a good way for the soccer ball factory to find out about this, because now you can make a ball made of 42 different, is it 42? 32, I think. Different pieces. So here we will have 12 pentagons, and I think there are 20 hexagons. And I have a question. If you want to make a ball that has, instead of 20 hexagons, it has 200 hexagons, how many pentagons will you need?
120.
120?
Did you say it was 12?
Yeah, so here you have 12 and 20 hexagons. Another answer over there? Yeah, so you just need 12. So it doesn't matter how many hexagons you have. You can have millions of hexagons. You just need 12 pentagons to make that an sphere. So if you have these pentagons distributed regularly in this structure, you can get a perfect sphere. So that's basically what viruses are doing. Fiedler also worked with tensegrity, and this, we have here another tensegridal structure. So these are just sticks and a rope. So you cut a rope any point here, and all these structures fall apart. And the same as here, it's just some sticks and ropes, and it's a flexible structure. If you press, then it comes back. So why I want to point out this, because Caspar and Klug also got some inspiration from these tensegridal structures. We usually think of a viral capsid as this rigid ball, but viruses also have elasticity as well. So in a virus, there are no ropes, but there are chemical interactions that are going to maintain the shape of the virus. So on one side we have the protein subunits of the capsid that are interacting with each other. On the other hand, we have the nucleic acid that is tightly packed inside, that's pushing this structure outward, and the compensation of all these tensions, and this is why it's called tensegrity, because of all the tensions, is going to keep the shape of the virus. So what's the theory of quasi-equivalence? How can you fit all these hexagons between the pentagons to have a sphere of any size? Well, what they came up with was with a planar representation of how you can arrange the different subunits in a capsid. So here, let's look at the background of this image. We have a grid of hexagons, like the ones I was showing before, and then we can connect each of the centers of these hexagons by lines to form triangles. So in a virus, each of these blue circles would be a different protein. Now we take an icosahedron, and we're just going to look at one of the sides of this icosahedron. So the side of the icosahedron is a triangle, and then we can make triangles based on this grid. So let's start by the simplest case. This one, for example, has the triangle of the icosahedron is just a triangle of this grid, and then you have in each of these sides you have 3 different subunits, and then this is going to be a particle with a total of 60 proteins. So this is like the fictional virus that I showed before with the hands. So we have in the 3 axes of symmetry, we have 3 different monomers. But let's turn now to build different sizes of particles in different ways. So this would be the next one in complexity. This one, the triangle, and this is called triangulation because of the different ways you can put the triangle in this grid. Here we have this orientation in the grid, and now what they came up with this formula that is going to give you a number that is called the triangulation number. So to know what number is for a particular vital capsid, you can use vectors to navigate from one vertex of the icosahedron, that would be the pentamer, to the next one. So for example, here we have this arrangement, and we can use vector this way. So now we are navigating along this triangle. So we are going to pass through the center of each of these hexagons, or hexamers, if we see it in a molecular way. So we would go one in one direction and one in the other direction to find the next pentamer. So if we apply this formula, we can get that the T number is H, the square H, so H would be 1, and K would be 1 as well, so it would be 1 times 1 is 1, 1 times 1 is 1, plus 1 times 1, 1 plus 1 plus 1 would be 3. So what is this number telling us? Well, it's telling us how these different subunits are arranged. It's going to tell us how many different, how many proteins we're going to find in a single vital particle, and it can also give us a sense of the size of the vital particle. So if the T is 3, like we see here, we just need to multiply that by 60 to get the number of proteins that are gonna be forming that capsid. So how many, if we have a T3 capsid, how many subunits would we have? 60 times 3, 180. Why is it called quasi-equivalence? Well, this wouldn't be quasi-equivalent, because as we saw before, each of the subunits in this capsid are going to be interacting with each other in the same way, but as you move over in complexity, you're going to have different interactions. So these subunits are going to interact with each other to form a pentamer. The other ones are going to interact with each other to form an hexamer. So that's how Fuller also built this geodesic. All the triangles are not exactly the same. They are changed in a way that they can all fit into a sphere. So that's basically what viruses are doing as well. So let's see a higher T number here, for example. Here we have, so we navigate through the center of each of these hexamers, and we have 1 and 2 to one side and 1 to another side. So what would the T number be for this one? So it would be 2 times 2, it would be 4. 4 times 1, sorry, 2 times 1 would be 2, and then 1 times 1 would be 1, so 4 plus 2 plus 1 would be 7. So we have these a virus in which the side of the icosahedron looks like this. We're going to have a particle that's going to have 60 times 7 number of subunits. I don't know how much that would be, 490?
490.
So viruses play with the different arrangements that you can have on this grid. So you can have different T numbers are represented here depending on how you play with this triangulation. So I know this is hard to understand, it's harder to explain, so please ask me questions. I don't know if this is clear. Well, yeah.
How did you get to the, I'm still trying to figure out how to get the number of proteins. Like, for instance, on the simplest one where the T number is 1, and there's 60 subunits, 60 capsomeres.
Yeah.
Is that because in those little circles are the subunits?
Yeah, so the circle would be the subunits. So
there's 60 of those on that simplest thing.
yeah, because you have an icosahedron that has 20 sides, and if each side has 3, then you have a total of 60. So what this formula is giving you is basically how many. It's not exactly how many subunits are on one side. But it's, I think it's like how many subunits you have like in a third of the side or something like that. So this would be 3, yeah, that makes sense. So you would fit 3 here, 3 and 3, and then 3, 9 times 20, 180, yeah, something like that. So the T number multiplied by 3 will give you how many units you have on one side, and you multiply that by 12, you get how many you have in the whole capsid. So let's look at some examples to see it more clearly. So I found this in the webpage of Protein Data Bank, and I found it very useful to see these triangulation numbers and how this theory of quasi-equivalence work. And they have origami that you can print and then you can fold as the ones that we have here. So this would be the simplest example. Is a T1 capsid. What you get in each side, you get 3 subunits, and then you can fold and you can make satellite tobacco necrosis virus that would be this big. And we're going to see that as we are trying to build viruses with a higher T number, the area that we cover is going to be larger. So we're going to make larger capsids as well. So this is T3. And the example here is tomato bushy stanch virus. And I actually have this virus here. And we can also see that when in the origami, the places where we have these corner here are the pentamers, and this is just the space that that would be the capsomer that is missing in the pentamer. There's not an hexamer, and that's what's going to give us allow us to close that into an icosahedron. So here we We can see what the T number is. So we navigate 1 here and 1 to the other, and we have seen that that's a T3. And what the color means here is how different each of these subunits are going to be to each other. So this is the same protein, but the same protein is adapting different structures to fill the space in a different molecular environment. So it's not going to be the same how the protein is going to be looking like in a pentamer and comparing to the hexamer. So I'm going to pass around a 3D model of TBSV. The cool thing about this model is that you can look in the inside as well, and you can see how different the pentamers are to the hexamers, and you can see how different they are. Then this one is a T4. So in the T4, if we look at the side of the icosahedron, we don't see an hexamer here, and sometimes it's hard to find these, to figure out this triangulation number. And it's because the hexagon is right here, hiding behind this panel of this one. So you have 1 and 2 on one side, 0 to the other side. So that would be 2 times 2 is 4. Oh, another thing that the T number is telling you, so I said that it's telling you how the different proteins are gonna arrange with each other, how many proteins are gonna be in a capsid, but it's also telling you how many different conformations the same protein is going to have within the same capsid. So if we look at this, we can see there is a red, pinkish one, an orange one, and a yellow one. These are the 3 different conformations that the virus is going to have. Then this one you have 4, so you have the pink, the orange, then you have light orange, and then you have yellow. This one is the T7, and then here you have 1, 2, 3, 4, 5. I actually don't see the 7 different conformations. Anyway, this would be a T7, like bacteriophage HK97. And we are gaining size and complexity. And then what happens if these pentamers are not distributed equally or regularly? Then we can make funky shapes like HIV, for example. So we just, wherever we have the pentamers, we are adding the curvature there. So in this cone, for example, we would have some pentamers here, but this looks more flat. There are no pentamers here. And as soon as we put pentamers, we get curvature there. These are just the images of the origami that you can make with these patterns. So feel free to take a look at any of these models, try to figure out what the triangulation number is. I'm gonna see if we understand this. So what would you say the T number is for this virus in particular? So first thing we want to do is find the pentamers. Can we see the pentamers? So here we have the pentamers. This would be the side of the icosahedron. Any answer? Okay, let's make it more easy. So now we have identified the pentamers. Now we have to navigate from pentamer to pentamer through the hexamers. So we're going to go through the center of each of the hexamers to find the next pentamer. So we move one this direction, next one, and then one to the other side. Then this is going to be a T7. Can we see this? Okay, let's see if we can see this one. So I'm not going to make you calculate the T number of this one, because this is actually T31, which is 1, 2, 3, 4, 5. 5. The square of 5 would be 25. And then 5 times 1 is 5. 25 plus 5 is 30. And then 1 times 1 is 1. So 30 plus 1 would be 31. This is actually a archaeovirus that was, which structure was discovered by Ken Stedman. And we see some decoration here as well in each of the pentamers. So if we look at the whole capsid, we can actually locate the pentamers very easily. So now we have a T31. How many subunits would make up this structure? Or if you don't have a calculator, how can we know? Multiply by 60. This would have 1,860 subunits forming this capsid. Yeah, any questions? It should be clear because I'm afraid this is important. Well, there. We back up 2 slides. 2 slides.
One slide back to the T7 number. Yeah. So I think this is for me just looking at differentiating H from K. So we're starting at a pentamer.
Yeah.
We're going into the center. Are those first the 2 purple arrows seem to be the H's and the red seems to be the K?
They're interchangeable. So it doesn't really matter.
Okay, that was my question.
Yeah. So of course there's exceptions as well. This is not a general rule. We have viruses like poliovirus, which is, if you look at this, if you just look at the structure and you find the pentamers, and then this would be the hexamer, that it actually looks like a kind of a triangular spiral. This would be what you think is a T3. But is it really a T3? Well, in poliovirus, there are 3 different protein subunits that are going to form the capsid. So we are not actually having the same protein that's going to have 3 different conformations to form the capsid. We directly have just 3 different capsid subunits. So that's what we talk. That's when we talk about pseudo-equivalence. So the way we would name the T number in this case would be with P before the 3. So the T number of poliovirus would be P3. Want to point out here as well the protein structure of the capsid protein. This is a beta viral that is called. It has a jelly roll type of fold, and this is something that is very pervasive in capsids. We find this type of fold in many different viral families, even if we cannot really find sequence similarity between them. Then we have adenovirus, and this is it has a T number of 25, and then the exons are formed by trimers. We can see also these fibers that are located where the pentamers are. This one is algal virus that has a membrane inside the virus, and it has a T number of P169. So it has a giant genome, so now it's trying to make a giant capsid. This one has 5,040 different subunits. I mean, they are not all different, sorry. But some of them are, and that's why there is a P here. And then we have mimivirus, ginormous virus. This one is Acanthamiba polyphaga mimivirus, and the T number is 1,179. So if you multiply that by 60, you get more than 70,000 subunits that are gonna arrange to form this capsid. Then we have viruses like this one, SV40. This one is formed just by one type of protein, so it's not it doesn't have what's called the pseudo-equivalence, and it has 360 subunits, which is very strange because there is no way to get a T number of 6 by triangulation. So what this virus is doing is it's actually fitting pentamers where we should have hexamers. So this would be what it would look like. So we would have pentamer here, here, and here, but these hexagons actually contain pentamers. There are some viruses that can form covalent bonds between the capsid subunits. So this is once all the capsid gets assembled, they actually form peptide bonds between the subunits, and this is what is going to confer this particle of a lot of resistance. So even though the particle is going to have a very thin shell, it's going to be very resistant. Okay, let's look at the helical symmetry. So some viruses can also just arrange all these capsid subunits in a helical fashion. So we have here the nucleic acid in red, and they're gonna sit on top of these proteins that are each of the capsid subunits, and they're gonna form a helix. So the cool thing about helical viruses is that all the subunits are gonna interact with each other in an identical fashion. It's not like the quasi-equivalent icosahedral structures where we find that the protein has to adopt different conformations. Here, the same protein is just looking the same all around, and it can also get as big as needed. So if the DNA is longer, you just need to add more subunits. The problem is that the ends are open. And then we can also make a calculation of how many subunits we have in these capsids. So in a similar way that we apply the formula with the icosahedral viruses, here we can calculate the pitch. And this is something that Francis Crick and his buddy put together right before Caspar and Clark came up with the theory of quasi-equivalence. So you just need to know how many subunits you're going to have per turn of this helix. There's going to be subunits per turn, and then also the displacement. So if we think of this helix as this planar grid that we then roll to each other, then we can have these subunits connecting with this one, and then the displacement would be 1, but this one connects with this one. The helix is going to come more this way. Instead of going like this, it's going to like open up, and you're going to have different stairs going up. So by multiplying how many subunits you have per turn, by the displacement, you get how many subunits you have in a particle. And then you have complex viruses like, sorry.
I have a question
Yeah.
about the helical virus. Is the inside hollow? Is it just?
Yeah, it's hollow.
Is it just like open, or is there?
So I think they don't have anything inside. Yeah, that's a good question. I don't know. Actually, don't know if that has a function or if.
Is there something that protects the ends? I mean, like some other capping protein or something?
There are some viruses that do. They have like different structures, and some of them don't. And I actually not sure what consequences that might have. I don't know if they are more susceptible to degradation. I don't know. I guess you can ask them. Let's say, and then we can find complex viruses that have both. So here, for example, this phage have a giant head, has a T number of 27, and then it has a tail that has helical symmetry, and this is contracted. And here we can see how we can have different displacements. As I was telling before, so if we follow one line of subunits that are purple, they're going to come like 1, 2, 3, 4, 5, 6 after. So here we'll have a displacement of 6, and if we have 10 subunits per turn, then we will have 60 subunits. I'm sure there are more, but so I guess the class is over. I'm sorry I couldn't go through the assembly stuff because I made a really cool toy that I wanted to show, but
We've got 3 minutes.
yeah. Made a video in case it didn't work, but I hope I can make it work. So while I prepare it, let's try to get to this link here. Oh. Sorry about that. So I made this kind of capsid in which we have the pentamers, the different pentamers. I put magnets that are gonna recreate the chemical interactions that put the different pentamers together. And this is just to illustrate how capsids can self-assemble. So we have here as well some genetic material. So the capsid proteins are also interacting with the nucleic acid. Some of them have basic residues facing the inside of the capsid, so they can interact with the nucleic acid. So I put some Velcro to recreate that type of interaction. So we can put here the difference of units separately. And this is a new DNA that I just got, so I don't know if it's gonna work, so I'm gonna race with myself with this video. So I have the subunits here, put the DNA inside. I think younger me is gonna win. Well, I didn't calculate very well the size of my DNA, so I didn't quite get it inside. But yeah, this is just to illustrate that some viruses, just by how they have, how evolution has shaped how the different amino acids are arranged in the protein that forms the capsid, can make the particles self-assemble in a certain environment, which is really cool. I don't know if you all saw that, but it works. Okay, thank you.